When I first started my own lab, which was actually about 35 years ago now, I was working not on telomeres, but on double-strand breaks. In other words, what happens in a cell when a chromosome gets broken. Cells don't like it when their DNA is broken, and they'll do just about anything to fix that up. And if they can't, they'll actually commit suicide to avoid causing further problems. Okay, so the way that we got into this is shown here. I was actually working on yeast at the time, doing yeast genetics. And with my friend Rod Rothstein, we'd made some initial kind of weird observations about what happens when you put circular pieces of DNA into yeast cells. That project was taken up by my first graduate student, Terry Orweaver, who's now a professor at the Whitehead Institute. What we found is that when you put in intact circles into a cell, you get very few transformants out. You get very few cells in which the circular DNA has integrated into a chromosome by recombining between related homologous segments of DNA. And the odd observation that we made is if you introduce a double-strand break, if you cut the DNA in that region of homology using a restriction enzyme, all of a sudden you get lots more recombinants. So it's very counterintuitive to us that this process would work better when you broke the DNA. You might expect, for example, that introducing a break here would just lead to degradation and disappearance of the DNA molecule, but instead we recovered thousands of times more recombinants. So that was technically useful, but we actually spent years and years trying to figure out exactly how that happened and what all the various biological implications were. But it turns out that one of the major pathways for the repair of double-strand breaks is this homologous recombination process, which, as you can see here, is pretty complicated. It involves a whole set of enzymes that do different processing reactions to the DNA. So there are nucleases that chew back from the site of the break, enzymes that catalyze strand invasion, DNA polymerases extend the invaded strand. You form these structures here called holiday junctions, and there are enzymes that resolve those to give you the final repaired DNA. And it turns out that cells or organisms actually use this pathway during meiosis. They cut their genomes up into hundreds of pieces as a way of stimulating recombination during meiosis. Okay, so basically everybody in my lab, me and all the students, we're all thinking about what cells do to repair broken DNA. And then in the summer of 1980, I went to a Gordon conference up in New Hampshire. And I met Liz Blackburn, and she gave a fantastic lecture on a really odd piece of DNA that she had been working on that came from this organism, which is Tetrahymena. The piece of DNA she was working with was linear, and she had found that at the ends of this linear piece of DNA, there were a whole bunch of repeats of this very simple sequence. And the thing that was most amazing to me, it really struck me, was that here you have a DNA end, but it's not acting like an end that cells want to repair or get rid of, right? It's acting like a chromosomal end, like a telomere. Now, the question was, however, since this came from Tetrahymena, which is shown over here, was this something special to Tetrahymena? Tetrahymena has a lot of weird aspects to its cell biology, including, for example, this macronucleus, which it generates by taking its regular chromosomes and chopping them up into thousands of pieces and amplifying them. And about the same time that we were doing our experiments, Tom Cech started to work on this because he thought this would be a great system for, we're finding the protein machinery that catalyzes splicing of introns. And so they started to work on it, and they purified and purified and purified, and in the end there were no proteins left. And this turned out to be the first example of an RNA molecule that catalyzes a chemical reaction, namely its own splicing. So that led to the Nobel Prize for Tom Cech, and years later this work led to the Nobel Prize for Liz and Carol Greider and for me. So a little 2 kilobase chunk of DNA from a really strange organism led to 2 amazing discoveries. And so I think that just highlights how important it is to not only to allow but to stimulate work on sort of odd systems here and there, because you never know where the interesting advances are going to come from. Okay, so here was the critical experiment. We had plasmids in my lab that had genes and origins of replication. So this DNA circle can replicate in yeast cells as a circle. What we did was to cut it to make it linear. If you put these linear molecules into yeast cells, they don't like it. There's nothing for the ends to recombine with. The DNA gets degraded, and you don't get anything out. The experiment then was to put the tetrahymena telomere DNA at each end, so that's shown here, introduce this DNA into yeast cells, and see what happened. And this was just one of those amazing experiments. I think this is probably the only time in my life this has happened to me, where I knew exactly what to expect if the experiment had worked. And in those days, we would run gels, and the DNA was radioactive, and we'd see what happened by exposing the gel to a piece of X-ray film, put it through this automatic developer, and then the film would come out. And usually you sort of hold the film up and sort of peer at the bands and try to figure out what's going on. But as it was coming out of the machine, I could tell that the experiment had worked. It was very exciting because this linear DNA gives a very characteristic pattern, because it's basically just one species on the gel. Okay, so it was very obvious right away the experiment had worked. These tetrahymena telomeres worked in yeast, so very, very divergent organisms. And so we knew right away, just from that, that the underlying biochemistry that took care of what was going on at the ends of chromosomes was highly conserved. Okay, so there were really 2 puzzles to figure out. So it had actually been known for many decades. This is going back to experiments of H.J. Mueller and Barbara McClintock in the 20s and 30s, that when chromosomes break, those new DNA ends are very reactive, and cells join them together. You get all kinds of chromosome abnormalities and so on. So there was something at regular chromosome ends that prevents this from happening. So that was one puzzle. The second puzzle had to do with replication. And so as soon as DNA polymerases were purified and people started to figure out how they worked, it became obvious that using that enzyme or that what was known about the enzymology, you couldn't fully replicate the ends of chromosomes. DNA polymerases always use a little RNA primer to get going. And so even if that was right at the end, the RNA would be degraded. You'd lose a little bit of DNA every time there was a cell cycle and another round of replication. So we know, since we're here, that our chromosomes don't get shorter every generation. So there had to be some mechanism to compensate for that problem. And so that was the other puzzle that we wanted to try to figure out. So I should say, the work that we did solved this question. And the question about what prevents bad things from happening from telomeric ends took about another 25 or 30 years to figure out. And that's mostly the work of Tizia DeLange, which I'll mention again a little later. Okay. Now, as I mentioned, I was working on recombination in my lab. Most of my lab was not doing telomere stuff. Everyone was, thinking about recombination. So when this question of how telomere biochemistry solved this incomplete replication, when that problem came up, I thought, I wanted the answer to be something involving recombination. Cells have a lot of enzymatic machinery that can do many different kinds of recombination reactions. And it seemed obvious that that machinery could be used to take care of the replication problem at telomeres. For example, by unequal sister chromatid exchange, you can take a short end and convert it into a long end. Or by using strand invasion and DNA polymerase, you could take a short 3' end, invade into a long end, and just add more DNA that way. My favorite model is actually shown here. We had some evidence from a few labs, which actually later turned out to be wrong. But there was some evidence suggesting that the ends might be joined together to make a hairpin like this. And so if you replicated this completely, you would just generate a structure like this. And this could immediately isomerize into a holiday junction, shown here. And then the recombination enzymes that resolve holiday junctions could cut along the dashed line, and you'd be right back to where you started with. This, I thought, was a very elegant way of getting complete replication of chromosomal ends. Okay, so unfortunately, all these beautiful models turned out to be completely wrong. And so there was the ugly facts that said that was wrong and pointed us, fortunately, in the right direction. And that is shown here. So the experiment was, or the observation was, I should say, that this is the DNA we put into yeast cells, right? Our vector plus tetrahymena, bits of tetrahymena telomere DNA. And these linear molecules would replicate. And so over many generations of growth of the yeast, it would continue to replicate. And by the time we were able to isolate enough of the DNA to look at, we noticed something really odd, which was the DNA had gotten bigger. It had picked up extra DNA from somewhere. So that was quite puzzling. And the real breakthrough came when we were able to get enough of this DNA to sequence it. And the sequencing showed that on the inside edge of the DNA, there were the repeats that came from tetrahymena, these simple GGGGTT repeats. But then added on to it was this extra sequence that was different. And it was a much more irregular GT-rich sequence. And it turned out later that this is the sequence that you find at the ends of normal yeast chromosomes. So this is yeast telomere DNA. This is tetrahymena telomere DNA. So we knew that this kind of structure could never happen by recombination. And so in the paper describing this result, we put forward the hypothesis that there should be a special enzyme that would synthesize more telomere DNA, and the sequence of that added telomere DNA could be different in different species. Okay. And so the very next year, of course, that enzyme telomerase was biochemically discovered by Carol Greider, who was at that time a graduate student in Liz Blackburn's lab at Berkeley. The enzyme has been studied ever since. We now know that it is a very complicated enzyme that has protein components and an RNA component. The protein component is a reverse transcriptase. So it reads a part of the RNA, which is acting as a template, and the reverse transcriptase synthesizes extra DNA, which it adds to the end of the telomere. The sequence that it synthesizes is determined by the sequence of the RNA template. And so different species have different sequences in the RNA template, and that determines the sequence of their telomere DNA. Okay. So the last major thing that we did in my lab in this whole area had to do with the genetics of telomerase and the effect on cells if you don't have enough telomerase to keep up with the telomere erosion that happens from incomplete replication. And so this is work that was done by Vicki Lundblad when she was a postdoc in the lab. And what Vicki did was to generate mutants that had defects in telomere maintenance. And so you can see, so the first mutant she called est1 for ever shorter telomeres. And you can see the effect over here on the DNA. So this band represents one of the chromosomal telomeres, and over time, over many generations, this band gets shorter, which you can see from it moving faster on the gel. The interesting thing, though, was the cellular phenotype. So with these mutant cells, when you start to grow them, initially they look just like regular cells, like normal wild-type cells. So after 25 generations, the colonies look normal. After almost 50 generations, they're starting to look a little raggedy around the edges. By 60 or 70 generations, they're obviously smaller. And by 80 or 90 generations, there are fewer very tiny colonies. At this point, there's just massive cell death. There's large amounts of chromosome loss. There are chromosome aberrations of all kinds happening. And so this delayed phenotype is essentially a senescence phenotype. It's as if the yeast are getting old because they can't maintain their telomere structure. And eventually the telomeres erode to the point that something bad starts to happen in the cells, and they start to die. Okay, so this finding, plus similar related work from Liz's lab, immediately sparked a lot of interest in the potential role of telomerase in both aging. So you can imagine as our cells go through generation after generation, eventually their telomeres get too short and you would have problems. And also interesting implications for cancer, which as immortal cells have to be able to maintain their chromosomes and their telomeres indefinitely. Okay, so at that time, the simple model for cancer would be that cells that didn't have telomerase would have a limited lifespan. They'd have a programmed number of divisions, cell divisions, that they could go through until their telomeres got too short, and then they would start to die. Whereas cells that had high levels of telomerase could, in principle, go on dividing forever, which is what tumor cells do. Okay, and so it very quickly turned out that most of our cells have very low levels of telomerase, at that time undetectable, but that over 90% of tumors reactivate the enzyme. Okay, so it's consistent with this simple model. Of course, things turned out to be more complicated than that. And really hundreds, maybe thousands of people have worked on telomerase in the ensuing 25 years. But I'll just highlight a little bit. A lot of the advances have come from work from Tizia DeLange, who showed that telomeres are not just naked DNA. They're actually in an interesting DNA structure called a T-loop, and they're protected by a large complex of proteins called shelterin, which prevents them from undergoing all these sort of bad reactions that broken DNA undergoes. And in parallel with that work from many, many labs, but especially Steve Elledge, who's also here at Harvard, showed that when you have a double-strand break, say in the middle of a chromosome, that initiates a whole set of signaling pathways and triggers various repair pathways, and if the repair pathways fail, it triggers cell death. Okay, so those advances together have led to this more complicated and in a way interesting picture of what goes on during the development of a tumor. Okay, so we can start off thinking if there's some tissue hyperplasia, excess cell division for whatever reason, or just some stem cell compartment where the cells have gone through enough generations that their telomeres are starting to get shorter and shorter. As soon as one telomere gets down to a critical length where it loses its protective structure and its shelterin complex, it will start to initiate this DNA damage response that you normally get from a broken chromosome. Okay, so when that happens, if the repair signaling pathways and machinery are intact, the cell recognizes that there's something wrong. It stops dividing, and then the cell will die. Okay, so no problem. That's fine. If there's a mutation, for example, in p53 or some of the other components of the repair pathways and signaling pathways, the cells will keep on dividing. Okay, and that's bad because then you start to get chromosomal abnormalities and mutations. But again, there's a safety valve, right? So if the cells still don't have telomerase, even though they're going through cell divisions and there's a lot of abnormalities, eventually the telomeres will all just get too short and the cells will die. So the final nail in the coffin, though, is if there's a mutation that turns telomerase back on. Now the cells can keep on dividing without limit, and then you have cancer. Okay, so that's sort of the current view of the relationship between telomerase and the development of a tumor, where at the beginning, low levels are harmful, but at the end, it's the high levels that are harmful. Okay, so at that point, I started to feel like first of all, there were going to be a lot of people coming into the field, and a lot of the work was going to be done in say mammalian systems, mouse genetics or working with human cells, and I wasn't really that thrilled about doing that, and so I started to look around and think about other things that I could get involved in, and so for a while we worked on some of the RNA enzymes that had been recently discovered. I mentioned the self-splicing intron, there were a few others. So we worked on that for a few years until we started to realize that, well, there weren't very many RNA enzymes known in nature. And we played with those, we worked with their biochemistry for a while. We were trying to make them do replication reactions. We were already interested in this idea that RNA copying itself could have been a key transition in the origin of life. We took that as far as we could, and it wasn't really working. So we started to think that actually what we should do is learn how to evolve our own ribozymes, RNA catalysts. And then maybe we could get molecules that did more what we wanted instead of what nature was offering us. And so that led to a sort of 10-year period of learning how to evolve through in vitro selection RNA and DNA and protein molecules that could do interesting and sometimes even useful things. Okay, so the basic idea is very simple. It's just like Darwinian evolution working on populations of organisms, except that here we're talking about populations of molecules, and we're the ones who are applying the selective pressure. Okay, so the general experiment is we start off with a very large library of variant sequences. So they can be completely random sequences, or they could be, say, a doped library, a sort of one basic sequence with a lot of variation put in. We then apply a selective pressure. So that could be anything as simple as selecting for sticking to a target, or much more complicated selections that are demanding a catalytic effect. We then collect the few survivors of that selective step and amplify them. And then we go around and around and around this cycle of selection and amplification until the population of molecules is taken over by molecules that do what we want. And at that point, we start to try to figure out what happened, analyze the sequences, look at what they're doing, solve structures, and so on. So this basic idea was worked out more or less independently at the same time in Jerry Joyce's lab and also in Larry Gold's lab, all with slightly different emphases. But we started off using this technology to actually isolate RNA molecules that could bind to different targets. And here's one of the early ones. This is a three-dimensional view of a small RNA molecule that folds up and generates a three-dimensional shape that is basically a little surface cleft that ATP just slots into very nicely. So it binds very, very specifically to this small target molecule. A little bit later, we figured out how to select for RNA molecules that could catalyze chemical reactions. And one of the most interesting was this one shown here, which is an RNA molecule that is a ligase. It will join together two other RNA molecules. And in fact, this ribozyme has actually been worked on for the subsequent 20 years, and it's been converted into an RNA polymerase, an RNA molecule that is an RNA polymerase. And what people were trying to do is to get it to be good enough to actually copy its own sequence. So you could have a self-replicating molecule. Okay. Later on in the 90s, we developed new methods that let us evolve protein sequences. And so what you're seeing here is a small non-biological. There we go. Protein domain. I mean, it looks just like any little protein domain that you would see, in any biological protein structure, but it's completely not biological. It was totally evolved in the lab from completely random sequences. Okay, so so I don't want to spend too much more. Great. Time on that. But after working on this kind of laboratory evolution for quite a few years, I started to get more and more curious as to how evolution got started, like all by itself on the early Earth, right? I mean, it's really easy when you have enzymes and a molecular biology lab, you can get this stuff to work in the lab. But it really, I started to get more and more puzzled by how Darwinian evolution really got up and running spontaneously. And of course, the corollary to that is the question of if life started here in that kind of way, could it have started other places? And so that question, I think, has really come to the fore recently from work over the last 2 decades by astronomers who've been discovering thousands of planets orbiting other stars. And so we know now that there are solar systems, millions, hundreds of millions of solar systems in our galaxy alone. Most of them are kind of different from ours, but some are similar, including this one, Kepler-62. So this is a movie put together by some people at NASA, and it shows that there are 3 planets that orbit kind of close in to its star, which is similar to our sun. But there are 2 planets that orbit a little bit further out in this green zone, the habitable zone, where liquid water would be a possibility on the surface of the planet. The inner planet, it's a little larger than Earth, and it would be a little hotter, but if it had enough of a cloud cover, it could be cool enough to actually have liquid water on the surface. The outer planet is probably more like Mars. It's probably cold and dry, but again, it's more massive, and it's possible that it has liquid water on the surface. So there are more and more examples like this, and the question everybody wants an answer to is, can there or is there life on any of these other planets? Okay. So another way of phrasing that is basically, so we know there are lots of environments out there that can support life, right? Whether they do depends on whether it's easy or hard for life to emerge from the chemistry of these planets when they're formed. A related question is, if there is life on any of these other planets, is it kind of similar to what we have here? Is it based on DNA and RNA and proteins, or does it use water as the solvent, or could it be really, really different? And I think both of these questions can be addressed by going into the lab and doing simple experiments and trying to figure out if there's an easy pathway that leads step by step from simple to more complicated chemistry to simple biology and then to more complicated biology. Okay, so you might wonder why it's taken so long and why we still don't really have an answer to this question. And I think one thing that held the field back for decades, actually, is that all the life that we're familiar with on this planet is really complicated. Right? It's complicated at a structural level. So this is just a picture of the cytoskeleton of a mammalian cell. But all cells have complicated internal structure, which is built on a really complicated underlying metabolism. Right? There are thousands of chemical reactions, thousands of enzymes that are catalyzing these transformations. And underlying this is a flow of information, which is also complicated. And the reason it's complicated is this flow of information from the archive in DNA through to the message and some of the machinery in RNA, and then on into proteins. It's a system where everything depends on everything else. Right? You can't make DNA without RNA and proteins and metabolism. You can't make RNA unless you have the DNA to encode it and the proteins to do the synthesis and the metabolites that feed into that, and so on. And so it took a long time to figure out how such a self-referential system could actually evolve. And there were all kinds of just crazy and bizarre theories that didn't really make any sense. And the answer came from what I mentioned at the beginning, which was Tom Cech's work discovering catalytic RNA, and also Sid Altman at the same time discovering a different ribozyme. Once it was realized that that molecule in the middle, RNA, could actually act like an enzyme, it became obvious that life could start in a much simpler way. Right? RNA can act as a genetic molecule, so you can inherit information by replicating RNA. We see that today with RNA viruses. But now, for the first time, we knew that RNA could act as an enzyme, could potentially carry out metabolic reactions. So it can do what proteins do, maybe not as well, but it can do it. It can do what DNA does, again, maybe not as well, but it can do it. So that seems like a great way to get life started. Only one biopolymer that we have to worry about instead of three. Okay, so the real question now is how do we get to cells that look like this? This is generally referred to as the RNA world hypothesis, the idea that there's an early phase in the evolution of life where the single biopolymer is RNA or some relative. Okay. All right, so to think about that, let's start by thinking about what kind of environments on the young Earth could support very, very primitive forms of cellular life. And so I've listed a few of the considerations up here. I think we need to think about environments where the organic building blocks of biology can form and accumulate to high concentrations. So I'm kind of dismissing the idea that life started in the oceans, where everything just gets diluted. We want to think about environments on land where the relevant building blocks can accumulate and get concentrated over long periods of time. We need sources of energy, and especially chemical energy, to drive the chemical transformations that we need to allow growth and division and replication to happen. There's a very particular feature of the environment that I think is very important and very interesting in constraining the possibilities, and that is that we don't want an environment that is, for example, always cold, right, where not much can happen. We don't want an environment that's always hot, where delicate molecules like RNA are just going to get totally destroyed. What we want is an environment that is, say, cool most of the time, but every now and then you have high temperature bursts. And that's particularly important for an aspect of RNA replication, which is that once you've formed a double helix, the easiest way to get the strands apart is by heating them up. But you only can do that very briefly, otherwise you'll destroy the RNA. Once the strands are apart, at least in principle, you can replicate them again. Okay, so what kind of environment has all these properties? And the answer is ponds or lakes. They could be volcanic craters or impact craters. that are geothermally active. So you have hydrothermal circulation, you have water circulating through. So let me talk a little bit about the chemistry that would feed into this. So one of the interesting theories, a theory that I like, and that's been pushed a lot by several people, but a lot by John Sutherland, is that the most interesting starting material for the building blocks of biology is cyanide. Turns out that cyanide is actually really easy to make in a lot of different ways in the atmosphere. So if you have an atmosphere that has a lot of nitrogen, CO2, water, maybe some of these other things, any kind of really a good source of a very high energy source such as shortwave UV or electric discharges, lightning, or even meteor impacts, will all heat these things up, basically rip the molecules apart into atoms. And when they come back together, they make very high energy compounds like cyanide. Okay, so now this will rain out of the atmosphere. If it goes into the oceans, I think that's not very interesting. It's just going to get hydrolyzed and disappear. But if it rains in to one of these shallow surface ponds where you have circulation through the rocks, what's going to happen is the cyanide is going to be trapped by reacting with metal ions. And it's going to form these metal cyanide complexes. And then salts of these complexes will precipitate. And those could accumulate over geological time. Okay, so this is giving you a huge reservoir of high energy starting materials. And when the geology changes, say there's lava flowing underneath, you get heated up, heating these metal cyanide complexes generates a whole stew of all of those things that you want to build the starting materials. So you'll generate a lot of derivatives of cyanide, like cyanogen and cyanamide, also acetylene and cyanoacetylene and so forth. And these compounds can react and make sugars and nucleobases, nucleotides, maybe even lipids. Okay, so there are environments that are like this in a geological sense in many places on the modern Earth. And what you're seeing here is a picture of Lake Yellowstone in the winter. So it looks very placid, and not to mention cold. But underneath, it's really amazing. There are all these vent structures, these conical structures, that are emitting streams of hot water, which can be very hot. And it's heated because of the water circulating through the rocks and getting heated by the magma underneath. Okay, so this is what we like to think of as the right kind of environment for getting life started, because now the environment can drive the cycle of growth and division. It can drive the primitive cell cycle at a time when there wasn't any evolved machinery to make the cell cycle work. Okay, so this is from a Scientific American article from a few years ago that I wrote with Alonso Ricardo. The basic idea is that you're in one of these ponds or shallow lakes. It's cold most of the time, and being cold or at least cool is really good for doing the RNA copying chemistry. Okay, but every now and then, these primitive cells or protocells would get caught up in one of these streams of hot water. It'd be transiently heated, almost to boiling. That will separate the strands of the replicated RNA molecules so that they can go through another generation of copying. It will also change the properties of the primitive cell membranes, allowing nutrients to rush in. But they don't stay hot, which would be bad. They immediately get swept out into the surrounding liquid where they're cooled off, and the chemistry of growth and replication can carry on again. Okay, so that's the kind of model that we're thinking of these days. And so in the last few minutes, I just want to show you a few things that we've done to try to build systems like this. So what the goal of my lab is now is to actually reconstruct this kind of process and actually show that it could work, at least in a laboratory setting. Okay, so here's a slightly more detailed model. Our view of a primitive cell is that it has some kind of cell membrane, not like the membrane of our cells or any modern cells, but simpler molecules, not a good barrier, but something that will let nutrients in and waste products out spontaneously, but that will keep the genetic material, the good stuff, trapped inside. Okay, the genetic material, we think, is something like RNA, maybe a slightly more primitive version of RNA, but it has to be replicated in a purely chemical reaction, because at the beginning there weren't any enzymes or even ribozymes around to catalyze that process. And so if you imagine this RNA replication going on at the same time that the cell membrane is growing and dividing, you would have a primitive cell cycle. And the idea is that by either starting with lots and lots of different sequences, or perhaps just because of the mistakes that are made during replication, eventually some sequence would occur that could not only just passively be replicated, but could actively catalyze some useful chemical reaction. Okay? Something that would actually be beneficial to the protocell that it was trapped inside. And as soon as that happened, as soon as you have a sequence that actually does something good for its host cell, that cell will start to take over the population. And that's the very beginnings of Darwinian evolution. That's what we want to see in our lab models. Okay. Once you have Darwinian evolution running, then I think it's much easier to imagine how you gradually evolve more and more complicated biochemical machinery, ultimately leading to us. Okay, so let me talk a little bit just about the membranes. The membranes we play with are made of fatty acids, like these. These spontaneously assemble into these really beautiful structures called vesicles or liposomes. So they're bilayer membranes. They're similar to cellular membranes, but they're much simpler and they're more dynamic. And basically these round vesicles, these spherical vesicles, grow into long filaments like this. And you're supposed to be able to see the process of growth, which is really kind of amazing and very biological looking. I'm sorry the movie's not playing. Then these filaments are very fragile and they divide easily to give you daughter vesicles. We have multiple ways of driving the growth and multiple ways of doing the division. So the end result is that we can go through cycles of growth and division in a variety of different ways, which is nice because it suggests there might be different kinds of environment that could support this cycle of growth and division. Okay, so in terms of the cell membrane, it actually looks like growth and division is not that difficult. There are easy ways of getting the environment to drive both growth and division. The thing that has been much more challenging is the chemical replication of the RNA. And this is something that people have been working on for roughly 50 years. At the beginning, the replication of RNA using simple chemistry looked like it might be pretty easy. And so a lot of the early work was done by the late Leslie Orgel. But by about 15 or 20 years ago, it turned out there was a whole list of problems that were basically preventing people from demonstrating RNA, chemical replication of RNA. So I won't go through it all, but basically the chemistry is too slow, it's not accurate enough. RNA is a kind of complicated polymer, and how you hook the building blocks together can occur in different ways. And so when you do it without enzymes, you get a very messy polymer with different kinds of connections. So that's looked like a big problem. If you could copy a strand and generate a duplex product, you can't get the strands apart. So it's a kind of dead end. And even if you could get the strands apart, they come back together again way too quickly. And then there are more and more problems. One of the problems that really bothered us was that metal ions both degrade RNA and are also required for catalyzing the copying chemistry. And those metal ions destroy the membranes that we're working with. Okay, so we couldn't really face this list of problems 10 years ago. So we started working on related nucleic acids, molecules where we thought the replication might be a little bit easier. So that was pretty interesting. But in the last few years, we've come back to thinking about RNA and trying to actually address these questions one at a time. And so I'm just going to very quickly mention a couple of recent advances. So one has to do with this backbone connectivity. So here's RNA as it's made biologically. It's always connected in exactly the same way, from the 3' through the phosphate to the 5' oxygen. But when you make RNA or copy RNA chemically, you get these incorrect linkages through the 2'. So the backbone is very messy. And I think everybody else assumed that when you don't have a uniform backbone, you're never going to get these molecules to fold up in a reproducible way. And so it'll be impossible to have, for example, good catalysts or even heritable catalysts. Well, it turned out that was just a preconception. No one had done the experiment, and we finally got around to testing it. It turns out that the RNA backbone is probably so flexible that it can actually tolerate this heterogeneity. It's not a huge problem. We went on to study the effect of this kind of incorrect linkage by determining the structure and looking at it in really great detail. And it basically makes very small changes that are just localized to the one place where the mistake is made. So here the linkage is occurring through this 2' position. So there's a distortion here, but it's compensated for by sort of opposite distortions in the flanking position. So overall, the structure is hardly affected at all. So that has really interesting implications because, so first of all, what we thought was a huge problem with RNA turns out to be not a problem. Second, it turns out it's actually a good thing because these 2', 5' linkages, these what we thought were incorrect linkages, actually make it easier to get the strands apart. So you don't have to heat the thing up so hot to separate the strands. So it actually solves a huge problem. And I think this is speculative, but there's an interesting possibility that comes from doing the chemistry and making these mistakes, if you will, which is that every copy is a little bit different. And some copies might fold out very well. They might make really good catalysts. But because they're folded up, they'd be hard to replicate. They'd be bad templates. Other copies that have these 2' linkages in different places might be terrible catalysts because they don't fold properly. But they would be good templates. So it's kind of a way of having your cake and eating it too, because of this very special property of RNA. And so we've gone from thinking that this backbone disorder is maybe a reason for thinking that life didn't start with RNA to now thinking that maybe this is why life actually did start with RNA. Okay, let me show you just one more short story, and this is this question of these metal ions, like magnesium ions, that are required for the copying chemistry, but which destroy these fatty acid membranes. And the effect is pretty obvious. So you can see it here. So here are some just normal fatty acid vesicles that contain a fluorescent dye. It's a very nice, smooth suspension. You throw in magnesium, and they just all precipitate on the side of the tube. It's just glop on the side of the tube. The membranes are destroyed. The contents leak out. There's really nothing left. And a couple of years ago, a graduate student in the lab, Kate Adamala, discovered that if you complex these metal ions with citric acid, the citric acid basically partly envelops the metal ion. It'll protect, that will protect the vesicles. And amazingly, it lets the RNA chemistry still go on. Okay, and that let us do an experiment that we'd wanted to do for many, many years, which is to copy RNA sequences inside these fatty acid vesicles. So we can load up vesicles with RNA molecules, primer template complex. We can add the necessary building blocks, the citrate, the magnesium, and the nucleotides from the outside. They'll cross the membrane spontaneously because these membranes are so permeable. and they'll copy the template sequence on the inside. Okay, so this shows, at least at a proof of principle level, that you can copy template sequences inside vesicles, which themselves can be replicating structures. So what we need to do, hopefully over the next few years, and hopefully in experiments that will be done by some of the lab members that I see here in the audience, is to just get this RNA copying chemistry to work well enough that we can go through indefinite cycles of replication. And at that point, I think we'll be in a position to actually drive the system for generation after generation and hopefully see the spontaneous emergence of new functions, okay, the beginnings of Darwinian evolution. And I think that if we can do that, that will be a huge step in understanding how life got started when our planet was very young. So all this work has been done by many talented graduate students and postdocs over the years, and support from many agencies. And so I thank all them, and thank you for your attention.
Are you willing to take a few questions?
Sure.
Maybe
yeah,
4 or 5 questions?
Happy to answer questions.
Great. Oh, surely.
Sure.
So, if it is easy to have this process happen and the protocells form, why would there be only one kind of life here?
Right. No, good question. So why is all the life that we see here basically, we can trace it back to a common origin? And well, the answer is that common origin of all extant life was already a very highly evolved organism. It was very far after the origin of life. Okay, so in fact, life conceivably could have started many times on the early planet, could have sprung into existence, gotten wiped out, or maybe started in many places and maybe they exchanged information. And I think there were probably at least hundreds of millions of years between the origin of life and what we now see as the last common ancestor, which has all the machinery of a bacterial cell. Yeah.
Based on the knowledge of the chemistry that you have now, is it conceivable that other small molecules or macromolecules other than RNA-like molecules could still support these types of reactions? Or is your sense now that this represents kind of a final common pathway at the core of mutants?
Right. So the question is, knowing what we know now about RNA and related molecules, could some other nucleic acid have played the role of RNA in our biology? Could we start life with something other than RNA? And I think that's still an open question. There are a lot of people working on that. Basically, the only way to answer that is experimentally to synthesize different nucleic acids, different genetic polymers. Think about how they, or whether they could have been made on the early Earth, and if they could have, do they have the right properties? We do know now that there are many different nucleic acids that can act as genetic materials. They show Watson-Crick base pairing, anti-parallel strands, all that kind of stuff. One of the more interesting ones is called TNA. It uses a sugar called threose. And TNA and a few other nucleic acids have been shown to be able to form functional structures. They can fold and make three-dimensional structures that can either bind targets or, I think, maybe in a couple of cases, catalyze reactions. So they're definitely candidates. It's not clear that they could have been made prebiotically, but I think this will get sorted out in the next 5 or 10 years.
I'll ask a question.
Okay.
If no one else has one. This is sort of a devil's advocate question, I guess. Have you been asked by, say, a patient or a member of the public, why is this being studied at Mass General, and what would you say?
Yeah, I do sometimes get asked that, and I guess part of the answer, at least, is that I'm in a department, the Department of Molecular Biology, which does a lot of basic research. This might be the most basic, but there are people doing, working on plants and all kinds of really things that you might think are very far away from medicine. But I think, as I tried to emphasize with our earlier work on telomeres, you can't really predict where the important advances are going to come from. I myself, I can't see any practical applications right now of the work that we're doing, but a lot of it's technology development. Some of the technology development that we did earlier for learning how to evolve molecules has turned out to be very useful. Maybe it would be great if something very, very practical comes out of this, but it might be 20 or 30 or 40 years. There are definitely common features of all forms of biological life that are puzzling and unexplained, and I think it would also be very satisfying to have some deeper understanding of why we are the way we are.
Okay.
All right,
Well,
thank you.
Thank you again.